So hello students, we are starting with the kingdom Monera. Now kingdom Monera, you know that it is the only kingdom of prokaryotes, and this is again a only kingdom with no nucleus. So we remember that in prokaryotes or in monerans, true nucleus is absent. So we say that they have no nucleus. Okay. So we deal with in this kingdom with the features, the characteristic of monerans, and then some of the bacteria which are included in this and their categorization. So if you start with this, first we are starting with the kingdom Monera, and in kingdom Monera, if we go according to the some of the characteristic with respect to NCERT, bacteria are the sole members of kingdom. So be remember, they are the sole members of kingdom Monera. Any other organisms are not found, not algae, not fungi, they cannot be found in this kingdom. Next, they are most abundant microorganism. If we take a spoon of soil, in this you can find millions of bacteria. So they are most abundant. They occur almost everywhere. You can find everywhere. Wherever you go, you will find bacteria. They can live in extreme habitat also. So we will deal with one group known as archaebacteria. And archaebacteria are those bacteria which survive in extreme habitat, extreme conditions. Now many of them live in or on other organisms as parasite also. We will study some of the heterotrophic bacteria as well. Now we are going to study structure in detail, so we will think that it is a little bit complex, but with respect to eukaryotes, or by comparing with the eukaryotes, we will find that bacteria have very simple structure and a very complex behavior. And this is the one of the important question, because in NEET they have asked this question in the form of blank. Now if we think about bacteria as a group, they show a most extensive metabolic diversity. They are most metabolically diverse organism, because there are different variants of different strains of bacteria are available, and they developed due to the different development and different evolution purposes. Now, so if we study monerans, first we should understand the structure. Of prokaryote or a moneran or a bacteria. Now this is the structure, a cell structure of prokaryote, and in this cell structure, if you visit, we should understand this diagram. In this diagram, you can see that there is a presence of a nucleoid. Thus what I have marked it as, it is a nucleoid. And nucleoid, it is not a true nucleus. So remember, it is a nucleoid. Why it is nucleoid? Because a nuclear envelope around it is absent. Nuclear envelope is absent. So because of this, it is known as false nucleus or xenophore or incipient nucleus. Now, besides that, you can find some of the polyribosome in the cytoplasm. We will discuss in detail this also. You will have a mesosome. You will find this mesosome also the invagination of plasma membrane. You find some of the lipid globules. Okay, then within there is a cytoplasm, and all these conditions, some of the RNAs are also present, then some glycogen is also present there. This is all found in the cytoplasm of this. But when we see the outer boundary, in outer boundary you can see that there is a presence of some of the respiratory enzymes which are associated with the plasma membrane. Now plasma membrane is the innermost, as you can see this, it is the innermost. Now outer to the plasma membrane is a presence of cell wall, and outer to cell wall you will find a presence of a glycocalyx. So this glycocalyx inner to that cell wall and then plasma membrane combinedly form a cell envelope. So we will deal with this in detail. Another to that, you will find a flagellum which is protruding outside. You will find some of the pilus also there. So this all things we see in this structure. So it is our prime necessary, or it is our duty to understand the bacteria in our detail. So to understand this detail, we should understand first some of the silent features of monerans. Now when we talk about the silent features of monerans are the unicellular. They are unicellular, they are made up of single cell. Now with unicellularity, later you can see that some colloidal forms are available and some multicellular prokaryotes are found, but common are unicellular. But one of the important point is that they are without nuclear membrane. They are without nuclear membrane, nucleolus, chromatin, and histone protein. All these things is absent. Because of this, we call a nucleoid, not a true nucleus. Then, if we talk about cell wall, that cell wall is made up of peptidoglycan. This is one of the fact-based question and direct-based question. You should remember that. But exceptions are archaebacteria and mycoplasma. In archaebacteria, peptidoglycan cell wall is not there, and in mycoplasma, cell wall is completely absent. So there is no think of a peptidoglycan. Later, membrane-bound organelles are absent. Then you can see a cyclosis is absent. Be remember, students. Cyclosis is nothing but a protoplasmic streaming. It is protoplasmic streaming. So, in this protoplasmic streaming, what is happening? Protoplasm is actually getting moved. So this type of movement of protoplasm is known as cyclosis, and the ribosome are of 70S type. Next to that, if we talk about some of the respiratory enzyme, as we have seen in diagram, respiratory enzymes are found associated with the plasma membrane. You can see in the backward diagram that there is a plasma membrane. With plasma membrane, some of the respiratory enzymes are involved. Then nucleoid, as we have discussed, or genophore. We remember nucleoid is also known as genophore, incipient nucleus, or prochromosome, and it is composed of naked DNA, RNA, and some of the non-histone proteins. Now, when we talk about reproduction, mainly they reproduce by asexual method, and in asexual method they perform a cell division, and cell division they perform a binary fission, and that binary fission is of amitotic type. This is one of the important question which was asked in NEET, and they lack spindle formation. So because of this, they have an absence of mitosis. Next, so we discuss this in detail. Then first we talk about the cell envelope. Now this cell envelope, when we are talking, cell envelope consists of glycocalyx, as we have studied. It is the outermost part of a cell envelope, and inner to glycocalyx you will find a cell wall and plasma membrane. Now this glycocalyx is represented by either a slime layer or capsule. We remember this glycocalyx is of two types, that is a slime layer or capsule. This slime layer protects the cell against dehiscence and a loss of nutrients. It protects a cell against desiccation and the loss of nutrition. And when a cell have a slime layer, they are smooth in appearance, they are rough in appearance. Actually, they are rough in appearance. But when they have a capsule outside these, that is made up of polysaccharide and D-glutamic acid, that polysaccharide and D-glutamic acid form a capsule, and it provides a gummy and sticky appearance to the progeny of the cells, property to the cell. So this gummy or sticky appearance is given, and due to this, the capsulated bacteria show a shiny colonies. Now, one of the important point which I am stating here, that you should remember that this capsule have a virulent nature. Which nature? They have a virulent nature. They have a virulent nature. So virulence in bacteria is given by this capsule. Then after glycocalyx, inner to that there is a presence of cell wall. Now cell wall of true bacteria, it is made up of peptidoglycan, as we have discussed, and it is also called a murine or mucopeptide. Now peptidoglycan is a polymer of alternating units of amino sugar. Means there are some sugars, that sugars are linked to each other, and their chains are linked to each other by the sum of the amino acid chains or peptide bonds. So their peptidoglycan is made up of N-acetyl glucosamine, as you can see here, that is NAG, and N-acetyl muraminic acid, which are joined together by a 1,4 linkages. And inner to cell wall you will find a plasma membrane, and plasma membrane is as phospholipid bilayer, as we will study in eukaryote as well. So they have a phospholipid bilayer structure. Now next, though this is a glycocalyx cell wall and cell membrane which is forming an outer boundary, that is a cell membrane. Now one point you should remember with this, that they have a single envelope system. They have a they have a single envelope system. Double envelope system is not that. Double envelope system is found in case of eukaryote, as they have a internal membranous system which cover the nucleus, which cover the other organelles, so that form another envelope system. So in eukaryote there is a double envelope system, but in case of prokaryote there is a single envelope system. Now when we move further, you will find some structures which are known as plasmid. Now what are plasmid? When there is a bacteria, in bacteria you find a double-stranded circular DNA. In spite of that, you will find a one double-stranded circular DNA which is separated from this, and these are known as plasmids. So plasmids are what? They are not actually, this is nucleoid, okay, but this is plasmid. So plasmids are what? Okay, they are a small extra chromosomal self-replicating double-stranded free naked DNA molecules. Means they are self-replicating, they can replicate by their own. They are extra chromosomal, means they are apart from the nucleoid, and they are self-replicating, they are circular as well, and they are double-stranded. Now the genes present on them have a no vital role. Be remember, they have a no vital role and in survival and growth of bacteria. But when plasmid temporarily, if this plasmid temporarily integrate with this nucleoid, okay, then this is known as episome. What it will be called? Episome. Now when we talk about types of bacteria are F plasmid and the R plasmid. Now F plasmid R forms a sex pilus, means if a bacteria have a F plasmid, then this bacteria will form a pilus, that is also known as pili, and it is responsible for a process of conjugation. So we will discuss conjugation in detail. Now another type of plasmid which are found in some of the bacteria, that is R plasmid, and that R plasmid provide a resistance gene. They have a resistance and they provide a resistance against some antibiotic like penicillin and the tetracycline. They provide a resistance against some of the antibiotic like penicillin and the tetracycline. So dear students, you should remember that this is the what, particularly we can say a nucleoid and double-stranded circular DNA, that is a plasmid. Because why I am drawing it separately, because we remember that is an important question. It is a important question. It was asked in NEET twice, so it is a star mark question which you should remember. Now next to that we talk about the polysome. What are polysome? So by hearing a term, poly means multiple, zome means what? Zome represents here ribosome. So ribosome usually lies scattered in the cytoplasm. They are found scattered in the cytoplasm, but sometimes they may form a small chain of 4 to 6 ribosomes. They form a 4 to 6 ribosome. Like you can see here, you can see this is a green color, that is mRNA, and over which this orange colored are ribosomes. Okay, so there are multiple ribosomes are attached over the single mRNA. That can be 4 to 6. Then it is known as polyribosome. Now this polyribosome is also known as polysome or ergasome. Okay, be remember, this is also important star mark question because it was asked earlier in NEET. So next we start with inclusion bodies. These inclusion bodies are the food storing bodies in case of prokaryotes or bacteria. Now in this we have seen that there is some of the glycogen granules. We have seen some of the glycogen granules, some of the lipid granules. This glycogen and lipid granules, they are the nothing but a type of inclusion bodies, and they carry out a storage of food. Now this is also important question, as in NEET they have asked this question twice, that storage bodies of prokaryote are known as, and the option was inclusion bodies, and another time they have asked this type of question, the inclusion bodies, what inclusion bodies do. So they store a food. Like this they have asked question, so it is an easy question but you should remember. Now next we talk about flagella. You know that when we talk about bacteria, from bacteria surface you can see some of the protruding structure which we call a flagella. Now these flagella are long, they are wavy and filamentous structure, as you can see here, and that protrude through the cell wall. So first important thing is that they arise from the cell wall. Now, what is the function of them? They provide a motility. They play a role in motility of bacteria for the movement. So, motile bacteria will have a flagella and non-motile will not have a flagella. Now, these are much thinner than the flagella or cilia of eukaryote. We remember one of the important points. They are thinner than the flagella or cilia of the eukaryotes. Now, we should focus somewhat on the structural part. So, when we see the structure which is given as here, in this structure you can see there is a cell wall and plasmamembrane. In plasmamembrane there are some rings you can see here that are a S and M ring, and above to that there is in a cell wall L and P ring. These are some of the rings which are present in this cell wall and cell membrane, and this ring combinedly form basal body. What they form? Basal body. Okay, now above to basal body you will find a here hook. Okay, and hook is connected to the filament. So if we talk about this plasma membrane and cell wall ring, the ring present in plasma membrane are known as proximal set, and the ring present in cell wall, that are P and L, are distal set. So it is made up of three things, that is basal body, hook and filament. Basal body has four rings, that is LPSM, but be remember one of the importance that only two rings S and M are present in gram positive bacteria. Gram positive are the purple or bluish bacteria which are stained with the crystal violet, and they have only S and M ring, means L and P ring which are present in cell wall which are absent in gram positive bacteria, while S and M ring are present in the plasma membrane, they forming a proximal, so all present in gram negative. Now hook is made up of different proteins and filament is made up of protein that is known as flagellin. So we remember that prokaryotes filament of flagella is made up of flagellin protein. That is the important question. We remember which protein? Flagellin. So that is the important question. Next, now we should talk about the arrangement of flagella, that is a flagellation types in bacteria. Now when you see there is an absence of flagella, that type of bacteria are known as atrichous. So atrichous means no flagella is there, means they are the non-motile bacteria. Now when there is a single flagella at a one end, it is known as monotrichous. When there is a two flagella at the opposite end, that are known as amphitrichous. When there is a group of flagella at one end, it is known as cephalotrichous. And when there is a group of flagella at both ends, as you can see here, there is a group of flagella at the both ends, but at one cephalotrichous, the group of flagella at the only one end. So this type is known as lophotrichous. And the last one is peritrichous. And peritrichous, you can see flagella is present all over the body. You can see here present all over the body. So that is known as peritrichous. And one of the known important example of peritrichous is E. coli. You know, E. coli is a Escherichia coli, and this Escherichia coli is the example of peritrichous bacteria. Okay, so remember this. You can get a question from this part, like the match the following question. Okay, so please remember this part. Now next, some of the other proteolytic structure is out of this flagella. They are pili and fimbriae. First you should focus in diagram what is pilis. So these are the structure which you can see here, that is a pilus, and the plural is pili. Some of the bristle like appearance structures you will see all over the body. Some of the bristle like appearance you can see here, that is the all over the body. Okay, so this bristle type structures you will see all over the body over the plasma membrane. They are known as fimbriae. These are hollow, non-helical. They are not helical. They are filamentous appendages projecting from the wall of the gram-negative bacteria. So this pili and fimbriae are usually found in gram-negative bacteria. These are thinner and shorter. They have more number than the flagella. First important thing, they can ask the question which are in more number. That flagella, fimbriae and pili are more in number, but flagella are few in number. These are made up of specific proteins called pili. So pili are made up of which one? Pili protein, and fimbriae are also particular Pillin protein. And this pili, that is a plural of pilus, help in conjugation. So we will discuss this one topic of conjugation in the sexual recombination process. So please students, we have discussed just flagella, which is made up of flagellin protein. Revise this point. Pili and fibria are also cell wall originated structures, but here pillin protein is that, and this pili help in conjugation. Now, so to look this fimbriae are like this, and pilus is like this. Now, to form this pilus, there should be a presence of F plasmid, because when F plasmid is present, F plasmid have a one gene that is known as F gene or F factor. Now this gene or F gene or F factor code for the pillin protein, and pillin protein accumulate in this part, and due to its accumulation there is a formation of pilus. So pili are formed when there is a presence of F plasmid. So please remember this concept. Now next, we talk about the different shape of bacteria. When we talk about different shape, you can see there are main 4 shape of bacteria. Okay. First is spheres that are known as cocci. The plural is coccus. When they are present in a group and they are 2, then it is known as diplococcus. When they are in chain, they are known as what? Streptococcus. You know, the strip means chain. So from the strip, the word is derived as streptococcus, means they are present in a chain. Then the rod-shaped bacteria, they are known as what? A particularly rod-shaped bacteria, they are known as bacilli. So you can find a chain of bacilli. Okay. In this cocci also you can see here there is a tetrad, there is a sarcina, a 3-dimensional structure, and when they are present like a grapes, then they are known as what? Staphylococci. Another one are the bacilli. Bacilli are rod shaped, and you can see here this is a spore forming structure. The spore former are found, they can be flagellated, and last one is the spirals. In spirals you will find a vibrio. In vibrio you can see the presence of flagella, and there is a spirilla. So these 4 types are found. So we go according to the NCERT. These are cocci, and these are cocci which are, you can see they are spherical in shape. They are spherical in shape or oval in shape, and they do not, they form a group, and they can be streptococci, staphylococci, as we have seen in this one. But flagella is absent, so they are non-motile. They are non-motile. But when we talk about bacillus, in bacillus they are rod-shaped. They are rod-shaped, and these rod-shaped bacteria are motile. They are motile. Now next to that you will find a spirilla. There are spiral shaped. Okay, and this spirilla are spiral shaped, and they have a flagella, as you can see here. So they are also motile. And last one is comma shaped. The vibrio, they are comma shaped, and this comma shaped vibrio do not form a group, but they have a flagella at this end, which are not shown here. So they are also motile. So here you can see 4 shape are there: spherical, rod shaped, that is a bacillus, then motile spirilla are there, and motile comma shaped vibrio are there. Now in this you can see a diagram of spore forming. The diagram, they can ask diagram based question from this. Okay. So this rod shaped motile or non-motile, we remember they can be non-motile as well as, because some bacillus do not have a flagella, so they can act as a what? Non-motile also, and spirilla always non-motile. Now if we want to talk out of this 4 shape, which is the most common shape, so we remember that bacillus are most common
shape. It is a most common shape of bacteria, and it is a star mark question. It was asked earlier so many times, so please remember this.